Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with ShahTareen Swati, and I'm here to deliver the lecture about the vectors. I got lots of requests to deliver the lecture about the vectors, their characteristics, and their types. So I will try my best to cover all these things and all your questions in my current lecture. So before starting my lecture, I have a very humble request to all of you to do just subscribe my channel, like my videos, share my videos. If you're sharing my videos, you are sharing the lecture and means you are sharing the knowledge. So keep watching, keep sharing knowledge with others, and do viral educational content. This is my request to all of you. So now I'm starting my lecture about the vector. In this lecture I will try my best to cover the cloning vector, and in my next lecture I will discuss about the expression vector. So first of all I will discuss about the cloning vector. What is basically the cloning vector and what is vector? Basically the vector, you can say that this is the carrier, this is the vehicle which is used for the transferring of the gene from one organism to another organism. In the recombinant DNA technology, in genetic engineering, so vectors naturally we get from different sources like viruses, bacteria, even from the eukaryotic yeast cell and human cell. So I will discuss all these things step by step, and sometimes the vectors are genetically engineered in the lab, and sometimes both cloning vector and expression vector combine together. So I will discuss all these things and all the characteristics with you people. So first of all, I am going to discuss about the cloning vector. What is basically the cloning vector? The vector which is used for the cloning purposes, and you are familiar with the cloning vector or cloning mechanism of cloning. I have discussed in detail about the recombinant DNA technology, DNA sequencing, and the cloning technology. So must watch my lectures about the cloning and the types of the cloning and so on. You will get a lot from these lectures. Okay. Now cloning vector, as you can see here, the cloning vector facilitate the amplification of a single copy of DNA molecule into many copies. Molecular gene cloning is difficult without the use of the cloning vector. So the first purpose of the cloning vector, as the name indicated, this is known as cloning vector. So it facilitates the amplification of the single copy of DNA molecule. Is it clear? So cloning vectors are used as the vehicle for the transport of the foreign genetic material into another cell. This foreign segment of the DNA is replicated and expressed using the machinery of the host organism. And the host organism may be the eukaryotic, may be the prokaryotic. It depends upon the situation and depends upon the condition. So it can be extracted from the viruses in the form of the plasmids, from the bacteria and other higher organism. Most of the cloning vectors are genetically engineered. It is selected based upon the size and the kind of the DNA segment to be cloned. So it is clear. This is the simplest introduction to the cloning vector. Now the general characteristics. What are the characteristics? Or you can say that the structure, or you can say that the organization of the cloning vector. The cloning vector should be small in size. Number one. It must have an origin of replication. It must have an origin of replication. It must have a compatible with the host organism. It must also be compatible with the host organism. First, small size is very important. Secondly, origin of replication. And you are familiar that there is no need to explain the origin of replication. The site which is responsible for the replication of the vector inside the host organism. It must be compatible with the host organism, like with the even we whether we are replicating or making the clone inside the prokaryotic cell or in the eukaryotic cell. So it must be compatible with that. It must possess a restriction sites, and restriction sites are those sites which are cut by the restriction enzymes. So restriction enzymes, which is used for the cutting of the gene of interest as well as for the cutting of the vector. So it must possess a restriction sites. The introduction of the donor fragment must not intervene with the self-replicating property of the cloning vector. This is very important. If the cloning vector donor fragment interfere with the self-replicating property of the cloning vector, so it will what? It create the problem. The cloning vector will not propagate properly. Is it clear? So a selectable marker, next one is that selectable marker, possibly an antibiotic resistant gene, must be present in the cloning vector. The function of the selective marker, you know that to identify the recombinant cells from the non-recombinant cells. Is it clear? It should be capable of working under the prokaryotic as well as the eukaryotic system. Sometimes we use the you can say that the host organism as the prokaryote as well as the eukaryote. So it should be capable of working under both conditions. The vector, a good vector, must possess the multiple cloning sites. Multiple cloning sites mean the polylinkers. And multiple cloning sites may be up to 25 sites in a cloning vector. Less than 25 are up to 25. So the multiple clone sites are also good for a good vector. So this is all about the some characteristics of the what cloning vector. And you can see here the cloning vector, all the characteristics like the restriction sites, like the selectable marker, and many more. Is it clear? Origin of replication and other. Okay. Now I'm going to discuss about the types of the vectors. I will discuss all the types one by one in my upcoming lectures. So the first you can see here the plasmid vector, then the bacteriophage vectors, the cosmids, the most important and most usable vectors. The plasmid, bacteriophage, phagmids, bacterial artificial chromosome, BARC, yeast artificial chromosome, YARC, human artificial chromosome, HAC, and retroviral vectors. I will discuss all these vectors one by one in my upcoming lecture. So must watch all my lectures about the vectors. Okay, these are the different types. I'm just giving you the brief view of all the types and the general characteristics. And now a little bit about the application of the cloning vector. As we are familiar with the application of the cloning vector. Cloning vectors are used in the for the cloning purposes, as well as it is used in the in making the genome DNA library, because they are cloning vectors and do the amplification of the gene, making the many copy of the gene, and in the recombinant DNA technology. I have discussed in detail about the recombinant DNA technology in my previous lectures, so must watch my lecture about the recombinant DNA technology. So cloning vector is used there. As you can see here, the cloning vector, the gene of interest inserted into the cloning vector, and that cloning vector with the inserted in means the recombinant. This is the recombinant DNA that is again put into the into the host cell for the amplification or for the cloning purposes. Similarly, for in making the gene library, this cloning vector is used. Similarly, cloning vector is used for the gene therapy. That is very important as a vehicle for carrying the gene of interest from one organism to another organism, or to replace the bad genes with the good genes. So these are different, you can say that applications in science and technology about the about the cloning vectors. So this is all about the cloning vector in a very simple way. I hope so the lecture will be clear upon you people. In my next lecture I will discuss about the expression vector, so must watch all my lectures related to the vectors and their types and their properties. I will be back. Till then, Allah Hafiz.